Okay, it's time, so let's get started. Good afternoon. My name is Minoru Ko from Department of Systems Medicine. Thank you for coming to Centennial lecture series commemorating 100-year history of Keio University School of Medicine. This school was founded in 1917, started from the friendship and trust between Yukichi Fukuzawa, a founder of Keio University, and also a founder of modern Japan in Meiji era, and Shibasaburo Kitasato, also a world-renowned microbiologist and immunologist, and also a leading physician scientist for infectious diseases. I'm sure many of you already participated in a big ceremony for Centennial celebration, which was done last Saturday. For this special occasion, it is our great honor to have Dr. David Schlessinger as a speaker. Dr. Schlessinger is currently an NIH Distinguished Investigator and has a long and stellar career as a scientist. He started his research career as an undergraduate student and became graduate student of Jim Watson's, first graduate student of Jim Watson's at Harvard in 1957. He then went on to do a postdoc research with Jacques Monod at the Pasteur Institute, Paris. He then became assistant professor and moved up to the rank of a professor at the Washington University, St. Louis. He established the first Genome Center and made a seminal contribution to the Human Genome Project. In 1997, he moved to the National Institute on Aging, National Institute of Health, NIH. He recently stepped down from the lab chief's position, but still continue to be very actively doing research and guiding a large group of scientists. As you can easily imagine, Dr. Schressinger's more than 60 years of careers as a research career is intertwined with the history of molecular biology from its inception. Moreover, he is one of the main figures who contributed and promote the molecular biology, its progress, by his outstanding contributions. We are very pleased to let you know that Dr. Schlessinger kindly accepted our invitation to give us 4 lectures, each of which highlights some aspect of molecular biology, together with his recent and latest accomplishment. With this introduction, I'd like to welcome Dr. Schlessinger. Please welcome Dr. Schlessinger.
Thank you, Dr. Ko, for your generous introduction. The extraordinary contributions of Keio medicine, both to clinical practice and to medical research, are well known worldwide. And it's a signal honor and privilege to be here with you and to speak with you in these lectures. As Dr. Ko indicated, the lectures will include new data, much of it presented publicly for the first time. But I will also attempt to give you some background to show you where some of the ideas and results come from. Today's lecture focuses on an attempt to optimize genetic analysis on a population cohort in order to help understand aging-related conditions and diseases. The project is called the SardiNIA Project. It's a collaborative venture between the residents of the Mediterranean island of Sardinia, the Sardi, and the National Institute on Aging. So it can be called SardiNIA or SardiNIA. To start with, let me recall to you something we're all familiar with now, that analyses of DNA variation are having a growing impact on medical practice. The primary discovery tool that's used for the genetic studies are genome-wide association studies, and these find many genes and gene variants that can affect either a trait, a phenotype, or a disease. Typically, just to recall the general principle, GWAS will look at variations at single bases in DNA. For example, some people might have a G at that position and others a C. Then we look at a trait, perhaps height, and GWAS asks whether taller people tend to have G at that position more often than C. And if so, we say that G is associated with height. This type of analysis has proceeded very rapidly. This is one of the first substantial ones that was done by a consortium of groups, including the group active in SardiNIA, looking at genetic variants associated with lipids, in this case, LDL cholesterol. And this is the type of representation that we've all grown increasingly to see in journals. It's called the locus zoom, in which we're looking across a stretch of DNA, and each of these dots represents a place where there's a genetic variant. We then look on the y-axis at the likelihood of one of those variants being associated with a trait, in this case, the level of cholesterol. So the likelihood expressed as the log of the base 10 of the probability that there's an association of one base version of DNA at that point is scattered around the baseline for most SNPs. Suddenly, we come to a point, one of the rare points in the genome, where there's a very high probability, on the order of 10 to the 13th to 1, that one of these SNPs, perhaps this top one, actually is showing association. And it then identifies a gene in which that variant is having an effect on cholesterol level. This has proceeded very rapidly in the field with a number of such studies. And the basic question that was asked from the start is, are the associated variants relevant for cardiovascular disease? We know that cholesterol is important. Do these variants have an effect? And the answer is resoundingly yes. For example, one of the first signals that was seen was in the gene PSCK9. And that has led to the development of a drug targeted to PSCK9, which is now in the clinics as a cholesterol-lowering drug. Now, why are we studying the particular population in SardiNIA? It has special properties that simplify the genetics in this type of analysis. SardiNIA is one of a few of what are called founder populations present at different places in the world. These are places where an original population came into the area and then expanded to the modern population with very little further in-migration. So the people are relatively interrelated. The SardiNIA is the most extreme and largest of these. The size of the population throughout history has been large enough to maintain high genetic variability. The original population that came in had excellent coverage of the total genetic variation in Europe. But in fact, looking at ancient DNA, we find that the original population, which settled there more than 15,000 years ago, was very much the same as the modern population. So they capture the great bulk of the genetic variation that was present in the original wave of settlement of Europe. At a time when most of Europe was under ice and glaciers, Sardinia was a paradise. People came there in large numbers. Then the land bridges that connected it to the continent of Europe were broken, and the population has remained relatively isolated since then. So there's relatively little heterogeneity on the island, and constant environment. The population are still living in the towns they're in. In the study that I'll describe, more than 95% of the people living in the towns we study had all 4 grandparents born in the study. That represents a true founder population. In effect, the modern population of 1.5 million Sardinians represent a single enormous family. Now, of course, the members of a family are different from one another, but the chances are that in a single family, the same genetic variants and genes will tip the balance toward a disease like diabetes in one individual in the family and in others. That is the reason that the genetics can be simplified. And furthermore, the size of the population is enough to give us cases and controls to study genetic factors in many diseases. Geneticists like 1.5 million myoses as starting material. So the population study called SardiNIA. This gentleman was one of the original participants, a picture of him at 102 years of age. And the motto of the study, translated into English, is "Your genetic endowment, a treasure you can share." The project takes place in a cluster of 4 towns in the east central area on the coast of Sardinia, an area surrounded by mountains on 3 sides and the sea. And there are 4 small towns in a single, very beautiful valley leading down to the sea. Again, it's a collaborative project between the Sardinian NIA geneticists. It was conceived of several years earlier and started in 2001 in this cluster of villages. The principal investigator in Sardinia is Francesco Cucca, a physician there, and I represent the equivalent at the NIA. Here are the key ingredients of the project. First, we've assessed about 800 quantitative traits. That is, by quantitative trait, we mean that every individual has a value for that trait, like a level of cholesterol. We've emphasized particularly immune parameters, as you'll see later in the talk, and a variety of anthropometric and hematological and cardiovascular measures. The study is done with an adequate number of participants to give us some power, both for epidemiology and for genetics. There are about 7,000 volunteers in the 4 towns, and these are grouped in roughly 1,000 family units. So we have parents, sometimes grandparents, sometimes 5 generations, and children as well. That facilitates the estimates of how much variation is genetic, that is the heritability, and also facilitates the analyses. In addition, we've separately collected now 4,000 multiple sclerosis cases and 1,500 type 1 diabetes cases. We'll come back to multiple sclerosis very soon. The participation of the population is enthusiastic. The population are very altruistic. They even participated in constructing the clinic that's used for the study. People in the villages brought in sofas and televisions and artwork for the rooms of the clinic. This is a longitudinal study so that we can do the same measures and even new measures roughly every 3 years. The 128 publications are now 145 most recently, and a third of those are based solely on the SardiNIA data. The large number of the others represent these large consortium efforts which gain power to detect small genetic effects across many populations. Most important, we have the full genetic information, the sequence of DNA, for all of the participants. There are 24 million bases in the 3 billion bases of our DNA where Sardinians show variation. This goes down to the level where there are so few people who have the variant that they wouldn't serve us for genetic studies. So we've recovered the full genetic variation that would be useful for genetic studies. Rather than show you some of the published GWAS efforts and other efforts, I thought it would be interesting to give you a feeling for how a new trait was introduced. We'd been studying a great many cardiovascular traits, including echocardiogram features, blood pressure, rate of blood flow, intima medial thickness, echocardiograms. But we also wanted to have a handle on the microvasculature, where we expect a great deal of action during aging. There is one place in the body where the microvasculature is exposed, and that's at the back of the eye on the retina. So we've studied this by looking at fundus photographs, now up to about 2,200 of the volunteers in the project, with an age range from 20 to 88 thus far. And looking at these photographs, it's possible to extract the segment by a segmentation program, the microvasculature. So that's the starting point for the analysis. We then define what we call microvascular features. This is a program done for us by one of our staff, Nikita Orlov. And first, it does the extraction of the vessel network, and then it begins to make measurements. For example, the number of junctions at which there's a split in the microvasculature, the length of the branches. You can see here purple spots where junctions have been assigned, and then the measurements are made, showing here in different colors for the size of the individual microvasculature elements. Now, ophthalmologists have not been slow to look at microvasculature. There is a literature on this, done mainly by direct observation or careful caliper measurements on individual photographs. So the samples have generally been small, but there are already links that have been suggested to several diseases. Looking at particularly tortuosity, which is the degree to which the microvasculature bends around in different elements, or junction analyses. And there have been indications that they might be correlation with hypertension or cardiovascular disease or increased risk of stroke. Again, very preliminary data for the most part. So we wanted to extend this using the automated extraction of the features in a population basis. And just to give you a feeling for the initial analyses, here we're looking at 11 traits. Most of them are tortuosity. A couple of them are related to number of junctions or length of segments. The 11 traits are across here, and they're doubled because we're looking on the top row at a younger group of individuals, and on the bottom row at an older group. And I'll just point out, just in the initial parts of this analysis, you can see here for these two traits related to junctions that there's a big difference between the analysis in the young and the old. These distributions represent the number or length of such junctions and the number of people who show it. So it's clear that the mean changes drastically and that the distribution even changes shape. So we know that there will be information here, and that gives us the first step toward findings. Again, each person will have a value for each of these traits. So we can look for correlations of tortuosity or junctions with cardiovascular measures, blood pressure and pulse wave velocity and so on. The ease of starting a new subject, a new topic like this, is great because all of the participants live within easy walking distance or a short ride from the clinic. So it's very easy to have them come back for their later visits and to introduce new traits. I'd like to spend most of the time that we have focusing on another feature of the project, which is that in addition to helping with traits, it can also resolve problems in the genetic analyses of factors in disease. Now, there are problems. This type of technology is incomplete. When you do an association test, you don't actually point to necessarily the right variant, the right single nucleotide polymorphism. An association usually points to a region of DNA, what we call a locus. And there are a number of DNA variants there. There are only a few examples of the successful identification of a particular DNA variant that's the one that underlies an association. And there are even fewer in which we have an understanding of the way that the variant affects a disease or a trait. And I'll show you a couple of those in the SardiNIA from the SardiNIA Project. Here's our first example. And this example relates to thalassemia. Thalassemia is seen in a belt of populations in all of which malaria was endemic until recently, and in some of them still there. Malaria has been the most powerful selective force in human evolution that we know. It's very simple. Malaria kills children. That's the most selective you can be. The result has been that at many places in the world where malaria is seen, there are secondary changes, mutations, leading to thalassemia, for example. Thalassemia is a defect in the level and nature of hemoglobin. So it's damaging to red cells and to metabolism, to oxygen delivery. Nevertheless, it's selected by malaria because thalassemia partially protects against malaria. So even though it's debilitating itself, malaria is worse. Now, the reason why SardiNIA has been especially useful in this case is that first, it was possible to find the first genetic basis for thalassemia in the Sardinian population. And second, it's been possible to use the population to find genes that moderate how vicious the thalassemia is. What I mean by that is that everywhere in the Mediterranean region, there are mutations to thalassemia, but these can be differential in their effects. In some of them, there's partial hemoglobin function left. In others, there's the persistence of a form of hemoglobin which is present in the fetus and usually turns off in adults, but persists in those individuals. So you can moderate the thalassemia. Now, because it's heterogeneous everywhere in the Mediterranean basin and elsewhere, you find many different thalassemia mutations. It's very hard to figure out how some people are escaping the drastic effects of the thalassemia. But in SardiNIA, although there are hundreds of variants around the world, there's only a single founder mutation because the founder population only had one mutation in it. That mutation is a nonsense mutation in the beta chain. And now, if we look at people who have that mutation but are not sick, because they all start from the same mutation, it's very likely they have the same moderating second mutation. So the GWAS was carried out, and this is one of the first most interesting findings from the study. We looked across the genome, And sure enough, there was a particular site at which, in contrast to the shaky background levels of all these different variants, we suddenly saw a particular variant strongly associated. The odds are 10 to the 35th to 1 that this is affecting the and moderating the thalassemia. It turns out that this variant falls in the BCL11A gene, and it is associated with the persistence of the fetal hemoglobin. So that explains why the people who have this variant, in addition to thalassemia, have their thalassemia moderated, but are still relatively resistant to malaria. I should point out that there were 30 years of work on the locus control region and other features of hemoglobin, of the hemoglobin system, trying to figure out what made the switch go from the fetal to adult hemoglobin. No one suspected that this gene might be involved, but genetics is agnostic. It points directly at that gene, and that gene turns out to be involved. Now, the implications of this association of BCL11A and the persistence of fetal hemoglobin are that the variant that we see accounts for most of the moderation of thalassemia. This is called in medical circles thalassemia intermedia. We did not have any cases of sickle cell disease, which is another hemoglobin modification that protects against malaria, seen mainly in Africa. But when we went to African populations and populations of African origin, it turned out that exactly the same BCL11A variant protects in part against sickle cell disease. So this gene becomes a target for some therapeutic approaches in SardiNIA and elsewhere. Now, the other example that I wanted to spend time on today is the immune system and immune system genetics, with special attention to autoimmune disease. So, first of all, rationale for immune genetic studies in humans. Thus far, what we know about the immune system and its regulation comes largely from work in mice, even in rats, and that is not always directly meaningful in humans. For example, in the 100 million years since we were mice, many of the forms of immune system cells, like many of the Treg family, were developed, so they don't even exist in the mouse system. Furthermore, systematic genome-wide association studies could systematically link naturally occurring genetic variation to specific phenotypes. I'll come back to this and explain exactly what I mean. Now, okay, we want to do GWAS for immune phenotypes, for example, the level of B cells. Why should we do those studies in SardiNIA? What's so special? Here we're looking at the prevalence of multiple sclerosis, the autoimmune disease, in Europe and in the rest, and in some Mediterranean countries. The high risk, represented by the red color, is seen in Scandinavia, to some extent in the UK. And there's a gradient from north to south. By the time you get to the Mediterranean, the number of cases per 100,000 is much lower, except for SardiNIA. In SardiNIA, we recently finished a complete estimate of prevalence on the island, and there are 350 cases per 100,000. That's more than twice the highest seen anywhere else in the world. So something is going on with autoimmunity in SardiNIA that's special. Here we see a similar plot for type 1 diabetes. Again, the well-characterized high levels in Scandinavia and levels at least as high in SardiNIA. So again, we're looking at a place where there's high level of autoimmune disease, and that increases the chances that we'll find something genetically related. Just to say as motivation why we might think this had happened, it's speculative, but we think again that this is related to the malarial selection. That in response to the challenge by the parasite, the residents of Sardinia went through a stage in which they had a convulsive effort to raise the level of the immune system. And they did, but that came with it, along with the increase of risk of autoimmune disease. So the benefits of studying immune traits and autoimmune diseases on the island. First, there's the availability that we already have of our SardiNIA cohort to use as controls. There are some cases there. And we also have a large collection of patients with the immune-related diseases and matched controls. Second, the diseases have this striking and anomalously high incidence in Sardinia. So we think we have more more ability to see genetic effects. And third, we can assess cellular subpopulations and their products efficiently in our cohort by having them come back and having blood drawn. So there are two strategies that can be used for this type of work, and I'll show you examples of both. In one, we search for genetic variants that control the levels of a particular kind of immune cell or of a cytokine, and then we look for coincident associations that affect, for the same very association that also affects the risk for an autoimmune disease. Again, I'll show you exactly what I mean by this, and it'll probably be clearer than the statement I just made. The other way that you can do it is a traditional way, just to compare cases and controls and look at the DNA of cases and the DNA of controls and see if there are genetic variants that are directly involved in risk. This can also be productive, although the disease itself may change the course of the and the nature of the people who are selected for in the study. So first, looking at the levels of immune cell types. And this is based on flow sorting of the immune system cells, as well as on direct determinations of cytokine levels in the serum. The work I'll describe is largely the work of the Sardinian team in this case, led by Francesco Cucca. And Eduardo Fiorello and Valerio Aru did most of the analyses that I'll show you. This paper, a couple of years ago, represents the first phase of this study. And it defines genetic variants that regulate the levels of many immune cell types. So again, what we do is we take immune cells, white cells from individuals. We separate them into specific cell types and do GWAS and look at associated genetic variants that are affecting the specific cell types and ask which of those, if any, are also correlated with an autoimmune disease. And the goal is, of course, to find potential therapeutic targets and help the patients ultimately. So what was initially measured? In the first part, the part that's been published, we measured 95 types of cells, and we looked at absolute levels of cells and also had other traits which were composed of the ratio of a cell to its parental cell or its grandparental cell during immune system development. These gave us a whole set of quantitative traits, and these were analyzed in all of the major classes of white cells. Leukocyte subpopulations, total B and T cells, and subtypes of T cells, dendritic cells, and maturation stages of T cells. So you begin to get hundreds of traits. And you see here the way in which the flow sorting was done. There are 4 panels of 6 antibodies each, and these are sufficient as fluorescent antibodies to separate successively all the cell types that we want to look at. Immune cells are very subject to change in their composition and numbers as soon as you take them out of the body. But having people come back to the clinic, individual white cells could contribute their white cells, and within 90 minutes, all of the cell types were separated and measured. The first thing that we asked about these is the general question that geneticists want to know. To what extent are the levels of cells heritable? How much of the level of B cells in individuals is genetic? And this was a surprise. It turned out that all of the cell types show substantial amounts of genetic influence on their levels. And some cell types, particularly a number of the Tregs, are 80 to 90% determined in their levels by genetics. I was surprised by that. I thought that the levels of immune system cells would just respond when you have an attack by an infection. Instead, it turns out that the levels of many of these types of cells are already fixed genetically and then are modulated further when you get the infection. This is the first GWAS that was done, and now we're looking across the entire genome from chromosome 1 to 22, and you see a huge background. This line determines the threshold above which there's certainty that the spots that you're seeing, the spikes here of individual variants, are truly associated. There were about 18 hits here that we could follow up in great detail. And these vary in strength. Many of them are very interesting. And as you'll see, some of them were seen in SardiNIA for the first time, had been missed by large consortia, but turn out to be important everywhere. So many DNA variants are associated with particular quantitative traits, and a number of those have also been spotted as associated with one or another immune-related disease. The difference that this analysis makes is that when we see a variant that's associated, for example, with the level of a particular type of T cell and has already been associated with lupus, for example, in GWAS, we now know a particular cell type that's affected by the variant and is likely to be a good candidate to start looking at functional effects of the variant. So this was in the first round, the Autoimmunity Association results and the Quantitative Traits Association results, and the overlap included a number of well-known features, some new ones. Right now, that's been extended to up to 140 cell types and a larger cohort of roughly 4,000 people, and there are now 57 genetic loci that are clearly associated in work that's now being written up. Much more work than can be worked on by the group in SardiNIA or at the NIA. So this becomes material for a large number of studies. Now, what about the other way of looking at autoimmune disease, the direct study of autoimmune disease? So this is where the cases and controls come from. There are 4 sites across the island where there are major hospitals that enroll the multiple sclerosis patients. And so we had 3,500 healthy controls recruited from across the island and 3,500 multiple sclerosis cases. Again, it was possible by using some tricks to cheaply get the full genetic analysis of all these individuals. So we know all of their variants in their DNA. And so we did a direct GWAS in cases and controls for multiple sclerosis. I'll show you some work in which a particular variant, in this case, has been identified as causal, and we know a great deal about its mechanism of action. That work just appeared in the 1st of May issue of the New England Journal of Medicine. This is actually, but again, this is the first time it's being presented publicly. And it was a collaboration mainly of the SardiNIA team and Laura Ida and Miriam Grosby at NIA, who carried out many of the functional experiments that you'll see. So what was this interesting variant that turned up? Here we're looking back at that first multiple sclerosis GWAS, and this was a signal that caught our eye. It's a significant gene in the TNF-SF13B gene. This gene is also called the BAF gene. It makes a product that's called the B-cell activating factor, which is required for the development of large numbers of B cells. And it had also been spotted in a GWAS that we did for lupus, as well as in multiple sclerosis here, but it had not been seen in GWAS of more than 20,000 cases of multiple sclerosis in a large consortium. We'll come back to that. But we had this signal, it was a new one, and so we followed it up. Now we look, we already know that this shows multiple sclerosis risk, and this is the locus zoom plot for it. And here we see the variant that's at the top, and then a number of others in a strange pattern. These are not real signals. They're pulled along because they're being processed in meiosis along with the true signal by linkage disequilibrium. Now here's a signal for B cells. Exactly the same SNP and the same pattern of other SNPs. Here's another one for the soluble B cell activating factor that's produced by the B cells, and it goes up with exactly the same SNP. And the level of monocytes show exactly the same pattern of all these SNPs with the same top one. This is what we mean by coincident associations. You can superimpose the GWAS profile that you get for each of these other traits with the one that we see for the disease. And that means that you're likely to find something out if you look at these cells in relation to BAF. A priori, you can see that since this gene is involved, the BAF B-cell activating factor is involved in determining levels of B cells, it's not surprising that it helps to determine the level of B cells. And since the BAF is the product of the B cells, okay, it's not so clear why it would affect monocytes. So some of the associations are immediately reasonable, others require further work. Now, what's the molecular action of the BAF variant? How do you get at that? Well, the first thing that was found is that when the BAF variant is in the DNA of individuals, it shortens the messenger RNA that's produced by the gene. So this is the transcript from the full message, of the full message. The 3' UTR is shorter when the BAF variant is present. And you can see that here in RNA-seq data from individuals who have 2 copies of the wild-type allele, 2 copies of the BAF allele, or heterozygotes containing 1 copy of each, with half the difference. The reason for this became evident when we looked at the nature of the variant. The variant is complex. It's not just a change in a base. It also involves an insertion of several nucleotides. And the result is that a new poly-A addition site is generated, which is extremely efficient. So the message that's made is shorter. Now, what effect does that have? The shorter BAF messenger RNA makes much more of the B-cell activating factor. So here we see the level that you get of the B-cell activating factor in people who have 2 copies of the BAF variant. And you see that in the wild type, the amount of BAF is only half the amount with heterozygotes in the middle. This is an enormous effect for a variant in GWAS. It's a doubling of the amount of product. So people with a long BAF mRNA only make half the amount of BAF of those with a short mRNA. How does that occur? Why should that happen? Well, the reason became evident when we looked further at the sequence nature of the DNA that was lost. It turned out that the part of the DNA that's lost in the shorter BAF variant form contains a binding site for a microRNA. The most microRNA that binds tightest is microRNA 15A. But the point is that when that microRNA binds to the 3' UTR, it reduces the translation yield of the message. So we should see here in direct measurements of the production of BAF in a cell line that makes the BAF product, that if we put in the microRNA, we can sharply reduce the level by the addition of the microRNA. So the molecular action of the BAF variant is that the shorter BAF mRNA escapes the microRNA inhibition. And in this case, then, we know not only the causal variant, but the mechanism of its action. Now, why was BAF variant not seen in multiple sclerosis before? Well, the reason is simple. The genetic variant is only present at low levels elsewhere, but it's selected to high levels in SardiNIA. And again, we think this is probably some kind of the part of the response to malaria. Nevertheless, the variant is in other populations. It has an appreciable, though lesser, effect on a population basis. So it doesn't account for as much of the risk of multiple sclerosis in a total population. But if you have the variant, it makes a big difference to you. The same great risk that it would in a Sardinian. There begins to be some clinical relevance of these BAF results. And in fact, there is an anti-BAF antibody, rituximab, which has been an approved and often effective therapy for lupus. So it's already approved against lupus. And the findings we made help to rationalize its effectiveness against this risk factor in autoimmune disease. You might then think that patients that have the BAF variant might benefit more from anti-BAF directed therapy, from rituximab, for example. But it's also possible that they might respond more poorly. And in fact, when we have looked at lupus patients who have higher basal levels of BAF, they had shorter clinical responses to rituximab. They had faster resurgence of their B cells. So it might be that they would have an opposite effect. The real implication in this case is that there should be further clinical studies in responder and non-responder patients who are treated with anti-BAF. You can stratify them by their BAFR status, and that could help advance the precision medicine, the individualized medicine for these autoimmune diseases. There's one other system that I'd like to mention briefly where there's an effect, there's interesting information from the SardiNIA Project. And that is the relation of autoimmunity to cancer. This is a more speculative comment that I wanted to leave you with. But in general, cancer is correlated with increased levels of chronic inflammation. However, people who are on immunosuppressive medication have increased risk of cancer development. And in a series of studies, this is one of the earliest, there's some much more decisive ones now, it's clear that MS patients show far less cancer prevalence than controls across the board for cancers. So that hints that there may be a connection between some features of autoimmunity and anti-cancer features. And there's a suggestion from the SardiNIA study of multiple sclerosis. One of the other genes that we had found, one of the other variants that had been discovered in SardiNIA, not seen in the consortium, but it turns out is also important for multiple sclerosis elsewhere. And these are variants within a gene called CBL-B, which is an immunoregulatory gene. Variants that lower its action are associated with multiple sclerosis. In this case, this is the locus zoom. You can see the most likely candidate SNP, the most likely candidate variant that's associated, and it's clearly within the gene. But this gene, although it was not known in human autoimmunity before, had been seen in mice because knockout mice for CBL-B reject spontaneous transfer of tumors. All the transplantable tumors are rejected by these knockout mice. You can see here that in heterozygotes you still have substantial killing, but the knockout mice tend to survive for long periods of time, rejecting the, in this case, TC1 cells. And this has been worked out by a number of investigators as working through particular CD8 plus cells. In fact, the CBL-B knockout mouse is one of the good mouse models for MS. So there's definitely a link and a suggestion of a possible connection to anti-cancer action. CBL-B regulates immune activation. The knockout mice are this MS model. And the mechanism is that in these cells, when you're lacking CBL-B, T cell activation doesn't require co-activation of CD28. It just requires the initial message from the presentation of the antigen. And in that case, the NK cells augment the inhibition of the cancer so that it becomes a therapeutic target. It's really analogous to PD-L1 receptor and other molecules that are brakes on the immune system. So again, in the SardiNIA, you eliminate the brake to get a huge immune response, and you increase the risk of autoimmunity, but apparently you decrease the risk of cancer. So to summarize the discussion of autoimmunity, the levels of most kinds of immune cells have a strong heritable component, and the GWAS analyses in SardiNIA find a very large number of genetic associations with the levels of various types of cells or cytokines. Some of these variants coincide with previously or newly detected autoimmune disease associations, and we now know the cell types where the variants and genes seem to act. So functional studies can begin more easily. In the case of BAFF-FAR, we know the molecular basis of the big increase in BAFF and the increase of B cell types of antibodies. And so you begin to have some clinical relevance supporting the utility of these immunogenetic studies in SardiNIA in health and disease. As a conclusion, I think for the messages that I'd like to leave you with from this talk are first that SardiNIA itself has provided a model for hematological and autoimmune disease, primarily because autoimmune disease is so prevalent there. It also has the founder nature that simplifies the genetics of many traits as well as the genetics of the autoimmune disease. But the genome is universal. Every population can help to analyze the common traits and diseases that are locally prevalent or are increased in prevalence in a local population for epidemiological genetic reasons. And of course, you have the Japanese centenarian study here at Keio and various GWAS studies, the Tohoku study here in Japan. Our genome is universal. We all share it, and all populations can contribute to genetic analyses of genetic pathology. Thank you for your attention.
Thank you, Dr. Schlessinger. So my name is Toru Takebayashi from the Department of Preventive Medicine and Public Health. So now open for the discussion. So is there any comment or questions? It's a great opportunity to
We may have tired out everyone. Late in the day.
Yep.
Well, I can ask you one. Do you think from a public health perspective that there are particular diseases in Japan or in regions of Japan that deserve more attention, both for epidemiology and genetics?
Yeah, in some infectious disease in the Kyushu area, HTLV1 infection, and then some disease, and then also in genetic disease, I don't know, but also in Tsuruoka, in the study area, also the marriage is some limited area, the next to kin, yeah. It used to say that something happened in that area, so maybe we can try to reach that area, then something.
So there's a hint of possible interesting projects. Yeah.
Well, thank you very much for really, really a great talk, and covers so much, very broad areas of research. So the question I want to ask is about this, your, not in details, but your sort of a comment and a speculation about the, why then Sardinians has this autoimmune disease, the higher prevalence of autoimmune disease like multiple sclerosis and then type 2 diabetes. And then your comment was, your thinking was sort of it's related to and caused by possibly malaria. Mm-hmm. And but then that poses some, of course, obvious question that what's happening in the other areas which are malaria is also prevalent?
Yeah.
Do they have also increased autoimmunity and some other diseases, I think, related to that?
Well, the general question is open now for speculation. It's hard to work on, but if you look at responses to malaria, you have sickle cell disease in some populations, thalassemia in others, so you do have a variety of responses. Why you would have particular ones in Sardinia, for example, it may depend on which plasmodium was there. There's several different ones, and the responses could be different. The major question is what's going on in Northern Europe? And there is no explanation currently for why there are high levels there. One thing that we've started to do is to compare, it's possible to look across the genome for signatures of selection. In other words, you can tell whether a variant in DNA has increased sharply in its level in a particular population or not by selection. And we can ask which alleles, which variants have increased in Scandinavia, which ones have increased in Sardinia, do they overlap, or do they touch on common pathways in the immune system? So there may be a way to get at mechanistically what's happening. But I've looked at the historical documentation, and there is no equivalent to malaria in Northern Europe. Nothing like plague or other things could explain it. So it's a good question, but no answer currently.
Thank you for your great talk. So I realize again, so quantitative trait, quantitative trait are important for GWAS analysis. And so you show the longitudinal data was also important. But I think so there is some limitation for the taking the data for the longitudinal data. So my question is, so the offspring, so their children, so who has half of the genetic information, so can we use as their the youngest? Can we use they as data for when he was young? Sorry.
Well, in the population studies, when you look at a trait, for example, level of cholesterol, you look at it. If you look at it across the age range, you normalize for the normal levels for people that age. So you have a way of accounting for the effect of age. And then, yes, you do see effects in parents and children. A portion of you have a trend that correlates with the alleles in certain genes. Of course, it's most evident when you have a mutation that leads to hypercholesterolemia, then it's already evident in the children. But even for the GWAS signals, is that fair, Jen? That okay?
Is there any? So SardiNIA is also famous at the dense population of the centenarian. So it's very famous for the longevity.
Yes.
So is there any study looking at the gene environment interaction, including diet and other lifestyle factors?
Well, the region that we're working in is the region which is famous for large numbers of centenarians. So we have collected DNA and information now for 300 people 90 and over. But the analyses are just beginning. My own feeling is, from looking at what we've done so far, is that, for example, if we look at kidney function, it declines with age, and it continues to decline with age in the very old people. So I think that in many ways, older people are like everyone else. They've just lived longer. And it could be that they've simply won the lottery on when terrible things happen. But there are many opinions in this field, and it remains to be seen. But we do have some material where we can start to do at least some studies.
So I appreciate all of you to be here. Thank you very much, Dr. Schlessinger. I was very moved your word. Every population contribute to study from the viewpoint of genetic analysis. It is very, very. Well, I was very moved. So this is a first step for our collaboration. Thank you very much for your good lecture. And thank you very much for to be here, all of you. Thank you very much.